Mitosis is a process of nuclear division by which cells create duplicate copies of themselves. The process is divided into 4 phases: prophase, metaphase, anaphase, and telophase and cytokinesis. Mitosis phase is preceded by interphase, when the cell conducts life processes and when the cell chromosome makes a copy of itself. During prophase, DNA condenses to form chromosomes. The nuclear membrane and nucleolus begin to break down. Centrosomes move towards opposite ends of the cell, and spindle fibers begin to form. In metaphase, the spindle fibers attach to the centromere and the chromosomes are moved to the equator of the cell. In anaphase, the spindle fibers shorten, splitting chromosome into chromatids and moving them toward a centrosome located at the pole. The unattached microtubules elongate, stretching the cell. During telophase and cytokinesis, the chromatids collect and spindle fibers disappear. The nuclear membrane and nucleolus begin to reform. At this point, this massive cell pinches and divides into 2, creating 2 daughter cells. Meiosis is the process by which cells create gametes. Like mitosis, meiosis is preceded by an interphase. DNA is replicated and each chromosome doubles, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. Meiosis 1, when a diploid cell with replicated DNA splits into 2 individual cells, and meiosis 2, when the 2 new diploid cells are split to produce 4 haploid cells. Meiosis 1 begins with prophase 1. Individual replicated chromosomes become visible, and each X-shaped chromosome consists of identical twin chromatids joined at their centromeres. Chromosomes that are similar in shape, size, and location pair up with one another, creating homologous chromosomes that form a tetrad. Chromatids of the tetrad may intertwine and exchange DNA, something known as crossing over. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In addition, the spindle fibers start to form and the nuclear envelope disintegrates. In metaphase 1, tetrads line up along the equator of the cell. Once they are lined up, the spindle fibers attach to each centromere. In anaphase 1, tetrads are split up into homologous chromosomes and are moved by the spindle fibers to the opposite ends of the cell. In telophase 1, the chromosomes collect and the spindle fibers disintegrate. The cell is divided into 2 and a new nucleus and nuclear envelope forms. After cytokinesis is complete, a second division, or meiosis 2, starts. This division is identical to mitosis. In prophase 2, centrosomes and the spindle fibers again start to form at the poles of the cell and the nuclear envelope disappears. In metaphase 2, the chromosomes line up along the equator and the spindle fibers attach to the centrosomes. In anaphase 2, the chromosome splits and the chromatids are pulled towards one end of the cell by the centrosomes. In telophase 2 and cytokinesis, the chromatids collect and the spindle fibers disappear. Once again, the cell pinches and divides into 2, and a new nucleus and nuclear envelope form. By the end of meiosis, there are now 4 haploid cells.